Welcome to the Wednesday afternoon lecture series. I am Michelle Antoine, Chief of the Section on Neurocircuits at NIAAA. It is my great pleasure to introduce today's distinguished speaker, Dr. Richard Chan. Dr. Richard Chan describes himself as a cardiac and neuronal functionalist, and we also see him as a pioneer in the study of voltage-gated calcium channels. His innovative work delves into the mechanisms and roles of these channels in synaptic vesicle fusion, cell signaling pathways, and their overarching impact on neurotransmitter release, synaptic transmission, and synaptic plasticity in both the healthy and diseased brain. For those watching remotely, we encourage you to participate in the question and answer session after today's presentation. You can do so by clicking on the button just below your videocast window that says Send Live Feedback. Click on that, type your name and question, and we'll relay this message to Dr. Chen at the end of his lecture. You can submit a question anytime, and I hope you do so. For those of you in the Lipset amphitheater today, you can use the mics on the left and right. We are also offering continuing medical education credits for this presentation. The CME code for today is 50095. That's 50095. So let's resume. Our esteemed speaker today is Dr. Richard Chan. He is a Druckenmiller Professor of Neuroscience, Chair of the Department of Physiology and Neuroscience. He earned his Bachelor of Science and Master's of Science degree in Electrical Engineering from the Massachusetts Institute of Technology. Subsequently, he was awarded a Rhodes Scholarship to pursue his Doctor of Philosophy degree in Biophysics at the University of Oxford, where he conducted research under the guidance of Professors Dennis Noble and Julian Jack. After Oxford, Dr. Chan joined Yale University School of Medicine as an assistant professor in the Department of Physiology, eventually attaining the rank of full professor within 9 years. After spending 18 years at Yale, he relocated to Stanford University in 1988, where he established the Department of Molecular and Cellular Physiology and served as its inaugural chairman. Here he also served as a co-director for the Stanford Brain Research Center. In 2011, Dr. Chan left Stanford as an emeritus faculty member and transitioned to the New York University Langone Medical Center, assuming the position of the inaugural director of the Neuroscience Institute. In 2015, he was named the inaugural scientific director of the Fresco Institute for Parkinson's and Movement Disorders at NYU. Dr. Chan has fearlessly forged a trail of groundbreaking research. His extensive research, documented in over 300 peer-reviewed journal articles, has significantly advanced our understanding of how neurotransmitters, drugs of abuse, and molecular alterations regulate calcium channels. Dr. Chan's contributions extend to elucidating the mechanisms that facilitate adaptation of synaptic strength or alterations in neuronal gene expression through changes in synaptic transmission and depolarization that prime brain networks for learning and memory functions. Additionally, his work has yielded valuable insights into the mechanisms that allow us to adapt to sudden or prolonged changes in activity levels. The implications of his research reverberate across diverse clinical domains, encompassing conditions such as Alzheimer's disease, autism, and neurological disorders like migraine, ataxia, dystonia. Moreover, his findings are pertinent to terminal conditions such as sudden unexpected death in childhood. Dr. Chan is also the co-author of the seminal textbook, Electric Current Flow in Excitable Cells, a fundamental resource for students studying biophysics. Dr. Chen's impressive list of accolades includes the Rhodes Scholarship, the Javits Investigator Award, Bernard Katz Award, the Cole Medal, the Kaiser Award, the Magnus Prize, the Cartwright Prize, the Axelrod Prize, the Gerard Prize, the Barnes Lectureship, the Annual Review Prize, the Distinguished John Burden Sanderson Prize Lecturer at the University of Oxford. Notably, he has been elected as a member of the National Academy of Medicine, the National Academy of Sciences, the Institute of Medicine, the American Academy of Arts and Sciences, and the Academia Sinica, which is the National Academy of the Republic of China. The title of Dr. Chen's talk today is Leveraging Genetics and Cell Signaling to Decipher Disorders of Excitability. Please welcome Dr. Chen.
And as Dr. Chen is approaching the podium, we had a little mix-up with the live feedback form. So for those people watching online, if you don't see it, maybe in a few minutes, reload your video cast system, and the button will be showing up in a few minutes. And the CME code, in case you misheard, was 50110. 50110. Thank you. All yours, Dr. Chen.
Thanks very much, Dr. Antoine, and thank you, Dr. Shore, and thanks to Diane Gomez for arranging this visit. It's a big pleasure for me to be here at NIH and to see folks well recovered from the pandemic and busily at work. I've met some wonderful people today, trainees and faculty, and I hope to meet some more during the rest of the day. I'd like to talk to you about a topic that is very relevant to NIH, and that is how we're going to take the discoveries from genetics and use them to understand cell signaling and therefore to decipher disorders of excitability, including epilepsy and autism and eventually schizophrenia. I have no disclosures to make, nor do members of my lab, and we have support from a number of organizations, including Brain Initiative, NINDS, NIMH, NIDA, NIGMS, but not specifically for the work on CBD that I'm going to tell you about. A number of different foundations have been very generous to us. In today's lecture, I'd like to show examples of how genetic insights prompt neurobiological approaches to function and pathophysiology. You can read this, I guess. Delineate which excitatory signaling molecules coordinate excitatory and inhibitory function. That's odd. Talk a little bit about sudden unexpected death in childhood, and eventually make some promissory notes about how we're going to take what we know about signaling proteins and apply it to a big disease like schizophrenia. Now, many people know that neurological and psychiatric disorders share genes and may even share therapeutic potential. In this slide from Joe Buxbaum's work, you can see a Venn diagram schematically depicting how the different diseases like schizophrenia, intellectual disability, and autism actually overlap with epilepsy in their genetic basis. Marcus, I'm having trouble finding the pointer. Oh, wait a second. Here we go. Can you see that pointer?
Yes, it's on the monitor, yes.
Okay. And indeed, Bill Catterall's lab came up with the idea that as one example of this kind of overlap, a medicine that was previously used for epilepsy, CBD or cannabidiol, was actually effective in treating a mouse model of autism. This is the Dravet mouse, a mouse that is deficient in NAV 1.1, and whose interneurons are not behaving properly. And this experiment by Nefi Stella and colleagues shows that if you use CBD at clinically effective doses in man, you can see an improvement in the social behavior of a mouse, bringing it up to sort of control levels as you increase the dose. So the possibility is that an anti-seizure medication might cross over and affect a social deficit that would traditionally be the province of psychiatrists. Now, I'd like to talk a bit about CBD because it rings bells with endocannabinoids, and it is a medicine that has been FDA approved. On one hand, we have the contrast between the psychoactive ingredient of cannabis, delta-9 THC, and cannabidiol, which is non-psychotropic. And that's an important thing for its use as a medicine. THC, as a number of people are studying, can have a number of different effects, including increasing hunger. Whereas, on the other hand, cannabidiol can sometimes counteract these negative side effects of delta-9. And finally, the CB1 receptor is the target of investigation of many people at NIH, including George Kounos, but the CBD has very low affinity at the CB1 receptor. So these chemicals are very close to each other. The key difference lies in this oxygen spanning link between two rings, which doesn't exist in CBD. Now, the medical motivations are abundant. CBD has been FDA approved following a series of double-blind, placebo-controlled clinical studies for its use in the treatment of Dravet syndrome, which is a form of intractable epilepsy where the subject has many, many seizures, and CBD caused a 40% reduction in seizure frequency. Likewise, in Lennox-Gastaut syndrome, another rare form of epilepsy, a similar modification was changed. So it's already been FDA approved. And on the street, many young people are using it for anything ranging from anxiety to insomnia without worrying so much about what dose they're using or how good the drug there is. So there's actually an experiment going on as we speak out on the street of people experimenting with this agent. And because it doesn't produce a high and is not illegal, there's a lot of testing going on. The question is, how does it actually work? And if we learn really how it works, can we apply it more intelligently and in a targeted way to affect diseases of the brain? This is work done by what I might call the CBD team, headed by Evan Rosenberg, an MD PhD student that is now doing his clinical training at Penn, and Simon Chamberlain, an electrophysiologist extraordinaire, along with Erica Nebbit, an undergrad who's gone on to medical school, Alejandra Sala, an MD, Guoling Tan, our lab manager, and Shahan Wang, who I'll talk about soon. Oren Davinsky was the man who did the clinical trials that led to FDA approval. And we were joined by Helen Sharfman, Swati Jain, and Yuri Vizhaki, and Drew Jones and Rebecca Rose. And we teamed up with a group at the University of Reading, whose work I'll shout out about, along with other people in the UK, together with GW Pharmaceuticals. Simon is going on to a faculty job at the University of Pittsburgh. Now, there are multiple candidates for CBD action, including the CB1 receptor or enzymes that are involved in affecting the production of AEA. It also can have actions on other ligand-gated channels, TRP channels, T-type calcium channels, but for today we'll focus on its action on one particular target, an orphan G protein coupled receptor called GPR55. This molecule lives in the membrane and responds to stimulation by its endogenous ligand called LPI. That agonism is blocked by CBD as a competitive antagonist. So this combination, a natural ligand, a receptor, and an antagonist, is the basis for the story I'm going to tell you in the first part of my talk. Why are we interested in these agents? Because of genetics. Genetics tells us, first of all, that LPI is interesting. That's because Joseph Gleason teamed up with groups in Pakistan and other countries in Asia and studied the metabolism of LPI. LPI is produced as a product of phosphatidylinositol, a very common lipid present in all your membranes and very important as a repository for arachidonic acid. When a cleavage is done by phospholipase A2, the arachidonyl moiety is clipped off, leaving you with a single acyl carbon chain leading to lysophosphatidyl'inositol, abbreviated LPI. And the synthetic enzyme for this is calcium dependent. So as a result of electrical activity in neurons, there is production of LPI. The reverse enzyme is called LPI acyltransferase, and it is the only enzyme known to convert LPI from this active form back into the PI reservoir. When you have mutations in this enzyme, and that was studied in highly inbred families in various countries, cousins marrying each other, just like in the royalty of the European capitals, the result is inactivating mutations that are doubled up on both alleles and therefore cause epilepsy, autism, and intellectual disability. So there was genetic evidence that the absence of breakdown or disposal of LPI could cause disease. However, the people who did the study interpreted it not in terms of the bad things that too much LPI would do, but rather the bad things that might happen if you had too little PI. And we're going to make the case that it's actually LPI that is the bad actor, and that LPI acts on its known receptor, GPR55, and that effect is antagonized by CBD. So genetics leads us to the ligand, and association through work of other people tells us about GPR55 and CBD. So if you use a knockout mouse kindly provided to us by Kenneth Mackey of Indiana University, you can see that GPR55 is absent in a region of the brain important for epilepsy and learning and memory called the hippocampus. So here we have staining for GPR55. Here we have only staining for the nucleus of the cell and no GPR55. And if you take mice and treat them with pentylene tetrazole to induce seizures, you find that there's a dose-dependent reduction in seizures in response to increasing amounts of CBD. But that dose dependence is gone in the GPR55 knockout, suggesting that GPR55 might have something to do with the seizures. What happens to neuronal function? We studied that in slices from the hippocampus that have the classical regions described by Romoni et al., the CA1 region, which is the target of synapses given off by CA3 pyramidal cells that send axons along the Schaffer collaterals to innervate the CA1 neurons. And when we stimulate those axons with an extracellular stimulus, we find that the stimulus is followed by action potentials that are recorded as extracellular currents, shown here, and that LPI increases the incidence of successful firing by about 50%, and an effect that disappears if you maintain exposure to LPI. So we can then look at the effect of CBD and show that the effect of the knockout is mimicked by the effect of CBD. So this is exactly what you would expect if there were a receptor, a G protein coupled receptor, and that CBD was the antagonist for it, So when you have CBD present, it's as if there was no receptor at all, and LPI is the endogenous ligand that increases the rate of action potentials by about 50%. This is hardly epilepsy, but it's done under mild conditions where we're looking to see that there's a physiological effect. Now, to understand how CBD had this action on transmission from the Schaffer collaterals to the CA1 neurons, we record quantal synaptic events from the CA1 neurons. These are called miniatures, or MEPSCs. They're studied under voltage clamps, so this reflects the inrush of sodium into the postsynaptic cell, triggered by glutamatergic transmission. And if we shrink these down and change the time scale to record over a minute rather than over 2 milliseconds, we see that there is spontaneous release of quanta from the presynaptic terminals, and that LPI greatly increases the frequency of these quanta, and the effect is reversible. And this is the time plot showing that on average there's something like a 70% increase in frequency, which is transient, and no change in the amplitude of the synaptic events. Evidently, the presynaptic terminal is releasing more quanta per unit period of time, and this leads to a transient increase in excitatory transmission. This is completely blocked by CBD, so fitting with the idea that it's blocking the receptor that triggers these downstream events, presumably on the presynaptic terminal. Previous work on this by Dmitry Rusakov's lab was confirmed by this slide, this figure. We went on to look at changes in inhibitory transmission. After all, this is only a transient change. Perhaps there was a change in inhibitory transmission that was more sustained. So we recorded inhibitory events like this, only not carried by sodium ions, but carried by chloride ions, triggered by the quantal release of GABA. And we found that in this case, the frequency events was not very much changed. What was mainly changed was the amplitude of the events. You can see all of these events are smaller. And that's shown here on a slower time scale over 10, 20 minutes. There is a progressive development in the effect. There's a slight decrease in frequency, and there's a very clear decrease in amplitude of the synaptic events. And this is also blocked by LPI. So evidently, the receptor is coupled to changes both in excitatory and inhibitory transmission. And in slides I decided to leave out, because it's in our paper and takes about 5 minutes to explain, we worked out the mechanism of this. We found that the number of GABA receptors in the postsynaptic cell, in the CA1 pyramidal cell, was decreased, and that this involved the gamma-2 subunit of the GABA receptor, a perfectly conventional modulation of inhibitory transmission. So Evan then went on to study evoked transmission by stimulating the same input that led to the evoked action potentials, recording from the pyramidal neuron, but now in response to an electrical input. And this showed results that were compatible with the recording of minis, namely LPI caused an increase in the strength of evoked transmission, and that was blocked by CBD and mimicked by knocking out the GPR55 receptor. There were also changes in paired pulse modulation, which are here for the aficionados who are interested in figuring out whether the effect is pre or postsynaptic. Not only did we see changes in excitatory transmission, but we were very pleased to see a change in dysynaptic inhibition, which is produced when the Schaffer collateral give off branches that innervate inhibitory neurons that in turn synapse onto the pyramidal cells. This is called dysynaptic inhibition, and we saw a big decrease in dysynaptic inhibition in response to CBD. So this is the decrease. It's something like a 60 or 70% decrease. And once again, this is prevented by blocking the receptors with CBD. It's mimicked by knocking out the receptor. And the changes that we see in amplitude are paralleled by changes in paired pulse modulation. There is a big increase in how much bigger the second response is to the first, indicating a recovery of release probability, and that once again is blocked by CBD and mimicked by the knockout. So evidently, release probability is going way down in response to LPI. So we worked out the mechanism of this, and I won't take you through it. It's very conventional receptor biochemistry, showing that the number and intensity of GABA receptors clusters has gone down, and that's because Gefrin has gone down and the gamma-2 subunit of the GABA receptor has gone down. I'm going to skip that to save time. Now, the key question is, how does this phenomenon relate to epilepsy? Is there something autocatalytic or regenerative about this? Well, we were very gratified to find that in response to a seizure, the concentration of GPR55 receptors was greatly increased, and that effect was blocked by CBD. So this is not just your common garden variety response where the receptors remain the same and the signaling do is what changes. Here, not only is the signaling changing, but also the number of receptors themselves are changing in a seizure-dependent and CBD-dependent way. This is the group data showing that in CA1 and CA3, and less prominently in dentate gyrus, there is a very clear increase in the level of receptor, which is reversed by CBD. So this means that there's an inherent positive feedback loop aspect to it, because the number of receptors that's causing this potentially damaging response is increased. And likewise, when we did experiments using RT-PCR against the mRNA that encodes the receptor, this also increased with the seizure and was decreased, reversed, and even more than reversed by the presence of CBD, as if there were an ambient level of LPI present in the slice even before CBD was applied, or an ambient level of the receptor. So how about the ligand? Is the ligand also influenced by the fact that you have a seizure? Well, we made an acute seizure with pentylene tetrazole, and one of the isoforms of LPI was actually significantly elevated by the seizure and more than completely reversed by the presence of CBD. This was done by mass spec HPLC measurements by Drew Jones and Rebecca Rose and our lipidomic score. And I want to point out that the isoform, particular isoform of LPI that is affected, is the 18-0 isoform, 18 carbons, 0 double bonds. This is the classic oleol form of LPI that was the very form that was increased in response to the knockout of the enzyme that reconverts this back into PI. So there is an alignment then between genetics and biochemistry. The biochemistry in the slice says that epileptic seizures reproduce the effect that is produced by the result in the consanguineous families. And I find this very satisfying because it provides some evidence that it's LPI and not PI that is the key factor underlying the manifestations of autism, epilepsy, and intellectual disability in those families. So to summarize what we've told you so far, LPI acts on a receptor. The receptor increases excitation and simultaneously turns down inhibition. That makes the neuron more prone to firing because the EPSPs are like an accelerator and the IPSPs are like a brake. And if you press down on the accelerator and let go of the brake, your car is going to go faster. That produces hyperactivity, and in turn, the hyperactivity causes an increase in both LPI and GPR55. And so every arrow in this loop is pointing upward, It's a positive feedback loop all the way down the line. And so this explains why epilepsy can be a runaway phenomena, and in fact, it can reoccur. And CBD comes along, blocks the receptor, prevents the effect of LPI from acting on GPR55, antagonizes the increased level of GPR55, decreases the level of excitation, decreases the seizures, prevents the hyperactivity, and basically puts out the fire, puts out the regenerative process. Or so we would claim. Let's seek some more evidence for this by seeing whether seizures change the level of GPR55 and the time course and the dynamics of that. So in these experiments performed by Helen Scharfman, Swati Jain, and their colleagues in our Department of Psychiatry, we are looking at the effect of a seizure produced by another epileptiform agent, kainate, which activates certain types of glutamate receptors and produces epilepsy very reliably, very hippocampally based. And if we induce a seizure by treatment with kainate, wait 48 hours, and then assess the level of the receptor using either PCR or immunocytochemistry, you can see a very clear increase in the level of GPR55, which is prevented if you give CBD at the same time that you give kainate. So 48 hours after a seizure, there's a trail, a biochemical trail, making the slice more susceptible to having another seizure because there's more GPR55 present. And this was true, once again, in the CA1 and CA3 regions. In both cases, there was a clear elevation in GPR55. Now, if it's the case that one seizure leaves a trail behind and leaves a predisposition for a second seizure, we should be able to come in with a second pulse of kainate and then look to see whether that produces a higher incidence of seizures. So we treated the tissue, the animals with a low dose of kainate, one not enough to produce a seizure. And nonetheless, we saw that we came in with a second dose of kainate. The second dose was effective 14 out of 15 times to produce a seizure to the second round. This is like paired pulse facilitation, 2 pulses of kainate. First one doesn't do much, second one does a lot, presumably because the after effect of the first seizure. Now, if we treat with CBD just before the second dose of kainate, then we prevent the seizures. That would be expected. But more importantly, if we treat with CBD during the first exposure to kainate, then the likelihood of seeing seizures is cut in half. That means that the trail left behind by the first seizure is dependent on whether CBD was present. And so this fits with the hypothesis that unchecked seizures beget further seizures. We tested this again by looking not at seizure susceptibility, but at excitatory minis to see whether the same type of effect could be observed there. We showed the effect of LPI in the absence of seizure in control tissues. It increased the frequency of minis. And then we compared that with what happened in tissue taken from mice that had been driven to seizures by lithium pilocarpine, yet another way of making seizures, and one that epileptologists swear by because it produces chronic seizures that will recur over and over again. And here you can see that the effect of LPI is greatly enhanced. It's more than doubled in size, and even after you wash off the LPI, the enhancement of mini frequency is sustained. So this is almost like LTP in that a synaptic input is prolonged in its efficacy. And so this is an indication at the cellular level that one seizure leaves behind a trail that makes it more likely to see a second seizure. And we saw a similar accentuation of the LPI-induced decrease in mini iPSC amplitude. Once again, that's postsynaptic. This effect is presynaptic. But in both cases, the chronic seizures make the effect bigger, not unexpectedly, because the number of GPR55 receptors is increased. And this experiment was done by Michael Bazelot, and we had help from Aston University doing control experiments that we could prevent this if the chronic seizures were antagonized by chronic treatment with CBD. So it all fits with the hypothesis, namely that CBD is a very clever drug in the way that it affects seizures. It prevents the positive feedback aspects of the circuit and takes out what is very characteristic of epilepsy, namely that one seizure can leave behind a tendency to have another seizure at a later time. Now, so far I focused on synaptic targets of CBD. How about ion channels, and are they involved? So we looked then, and this is Simon Chamberlain, looked at two major categories of neuron. We're not going down as fine as to all the various subtypes. We're just going to distinguish between fast spiking interneurons and regularly spiking interneurons that are characterized by being rich in the marker somatostatin. They have very different morphologies. The PV neurons are very often basket cells with cell bodies and lots of axonal ramifications in the pyramidal layer, where they act as a damper on pyramidal cell excitation. The somatostatin neurons have a different morphology, and together we can schematize them in talking about feed-forward inhibition, which occurs when the Schaffer collateral input fires a PV neuron, and that inhibits the pyramidal cell. So that's feed-forward inhibition. And then after the pyramidal cell fires, or if it fires, the somatostatin neuron is caused to release GABA far away from the cell body, transferring an input in one layer of the hippocampus to an inhibitory input in a different region of the hippocampus. And this is heavily studied by Chris McMahon, Ken Pelkey, and their colleagues right here in NIH, and they have gone on to even more types. But for now, this is enough to make the point I want to make, that the feed-forward inhibition is very effective in quelching a spike even before the chances occur, whereas the feedback inhibition has other purposes. So what happens when you treat with CBD to these forms of inhibition? Well, here's a more physiological input, a short train of action potentials at 50 hertz. So the action potentials are 20 milliseconds apart, and we're going to give 5 of them. So this is typical of a burst of firing often occurring in the hippocampus in response to systems neuroscience level activity. In the control, in this slice, there may be one spike, maybe 0 spikes, but not very many spikes. In the presence of CBD, all the spikes are completely gone. And on average, there is a very prominent change in the stimulus number dependence of whether there's an action potential or not. Excitability is greatly decreased in the pyramidal neuron. So why does this occur? Well, let's look using stimuli that are weak enough not to produce any spikes, so we can look at the compound synaptic potential that arises from excitatory transmission and inhibitory transmission, all occurring at the same time. In the absence of CBD, we see a gradual buildup of depolarization. This is very physiological. This is what would make the neuron reach threshold, only we're not quite hitting threshold here, so we can analyze the synaptic events. In response to CBD, the net depolarization is strongly decreased, consistent with the lowering of the number of action potentials. And this is group data showing the same thing. Now, what happens if we knock out the receptor? Well, in the absence of the receptor, this effect is completely gone. What happens if we mimic this with an artificial agent, not a phytocannabinoid like CBD, but an inhibitor of the GPR55 that's been engineered to be selective for the GPR55 receptor? That's called CID, and at a low concentration, this mimics the effect of CBD. In presence of CID, we apply CBD, and now CBD has no additional effect. So the effect of CBD has been occluded by the presence of CID, once again reconfirming the idea that this is due to the GPR55 receptor. Whether you knock out the receptor and eliminate the effect, or stimulate the receptor using CID and get the full effect, either way, it's very convincing evidence that this effect on excitability and synaptic transmission is GPR55 dependent. Now, the key experiment is to compare the results of excitatory transmission alone or excitatory transmission with GABA receptors blocked. So normally we have both glutamate receptors and GABA receptors. If we block GABA receptors, now the effect once again of CBD completely goes away. So that means that this was not an effect on excitatory transmission. It was an effect on inhibitory transmission that counteracted the effect of the excitatory transmission. So we see a complete buildup of the EPSPs without any interference from the IPSPs. So that tells us that the effect on the neuronal firing is actually mediated by the GABAergic system and not the glutamatergic system. So which neuron is responsible for this? The obvious possibility is that PV neurons, which mediate feed-forward inhibition, are involved. So to study this, we can now record from PV neurons. So this is not the spiking in the excitatory neurons. It's the spiking in the PV interneurons upstream of the excitatory neurons. And lo and behold, CBD greatly increases the firing, maybe threefold. So the PV neurons, instead of firing every once in a while, fire almost nearly all the time. And this effect is mimicked by CID, showing once again that it's due to GPR55. So now we have an effect of CBD on the inhibitory neuron itself, not only on the inhibitory neuron, but the very inhibitory neuron that is most strategically located and placed in the circuit to quelch action potential propagation. How about SST neurons? So when we apply CBD here, we see the exact opposite of what we saw for the PV neurons. Instead of these neurons firing more to increase inhibition, they are firing less. And that effect is also mimicked by CID. So the two interneuron types, the two main interneuron types are affected in opposite directions. What is this due to? Is it due to the excitatory transmission, or is it due to their intrinsic excitability? Because remember, we're firing spikes here. We're describing the probability of spikes. So this shows what happens if you depolarize using the patch clamp electrode, bypassing synaptic transmission to make the neuron fire spikes. Here's a fast spiking interneuron, and in response to CBD, the action potential frequency goes up significantly, particularly prominently at small current injections. So CBD enhances the intrinsic properties of firing of PV neurons. It has the opposite effect on SST neurons. So this is very clever because it's enhancing the excitability of the very neuron that is best positioned to mediate feed-forward inhibition. Now, we tested the effects of sodium channel activators on the hypothesis that this might involve enhancement of sodium currents. And the hypothesis would be that CBD could prevent an endogenous downregulation of sodium currents mediated by conventional signaling pathways studied by Catterall and Jim Surmeyer. And if there was a tonic amount of LPI downregulating the sodium current, then CBD, by interfering with the signal transduction, could make a bigger sodium current or more firing. And by looking at the compound EPSP, we're able to show that a synthesized stimulator of NAV 1.1, the sodium channel that's in the parvalbumin neurons, was able to mimic the effects of CBD and, in fact, completely occlude the effects of CBD. And this was mirrored by another compound, AA43279, once again mimicking and occluding the effect of CBD. So we think we are on to what the basic mechanism of how CBD might act, and We then tried to test what the timing of the input needed to be by doing an optogenetic experiment, expressing channel rhodopsin in PV neurons, and showing that we could mimic the effect of CBD without any drug at all by shining light on the terminals of the PV neurons onto the pyramidal cells. And you could see that regardless of whether you have weak stimulation or strong stimulation, the excitation of the feed-forward inhibition mimicked the effect of CBD. This was not produced if we shown a very general and time indiscriminate steady glow of light. Then we saw the exact opposite of this effect. And Scott Owen, who was working with the intramural director's son, relative, was the one who described this for PV neurons using oxytocin. The general light is doing what oxytocin does. It's the opposite of the acute light. So what you need in order to prevent the throughput of transmission is precisely timed activation of the PV neurons at the right place and at the right time, right as you would expect from feed-forward inhibition. So the conclusion is that CBD is a very clever drug. It dampens the propagation of high-frequency activity in the hippocampus by increasing PV interneuron firing and concomitantly decreasing the firing of SST interneurons, maintaining some sort of PV SST, I want to use the word balance. I don't want to use the word balance because it reminds me of EI balance. But anyway, it's some kind of harmony between the two types of interneurons. The interneuron that you need to fire is the one that's made to fire. And that dampens spike transmission via pyramidal cells, and the SST neurons are not allowed to inhibit the PV neurons, so that even makes the effect even stronger. We can mimic the effect by activating sodium channels, and they mimic and occlude CBD circuit action. And the timing is important because that's when you need the inhibitory input to prevent firing. And these experiments, which are done without added LPI, are really better suited to the neuropsychiatric indications that CBD might be used for, because the people who are taking CBD for whatever ails them, sometimes intelligently and sometimes maybe blindly, are not epileptic patients. They're just people working with the brain's own level of LPI. And so the virtue of having an endogenous system is that it's modulating the properties of the circuit and regulating how information gets transferred, and it provides a strategic target for CBD action. Now, let me check how I'm doing on time. And I'd like to segue to the last part of my talk. And believe me, there are not 50% more slides. There are only maybe 33% more slides, and I think I have enough time to do this.
So this might be a good moment. I'm over here, the other direction. Hi. I just wanted to announce, our send live feedback button is not working, so anyone watching it remotely, and there are a lot of people watching remotely, send email to the WALS office at od.nih.gov, WALS office at od.nih.gov. Thank you. Excuse me. Continue.
Okay. Thank you. We want all the feedback we can get, and we're happy to get it, whether it's in a chat or an email or any form. Email might be even better because I can pass it on to my colleagues who are always looking for ways of improving our work. So I described this vicious cycle, this positive feedback, where sometimes the ligand and always the receptor that triggers an increase in excitation and a decrease in inhibition is enhanced by hyperactivity, a positive feedback loop that can be interrupted by CBD. This is at the receptor synapse level, and at that circuit level, the number of spikes that can get through is enhanced by a great enhancement of feed-forward inhibition, a concomitant reduction in feedback inhibition, and together this decreases the net EPSP and decreases spike throughput. So both at the synaptic level and at the circuit level, neuronal firing level, CBD is a very effective agent in modifying impulse, traffic, and information flow through the brain. I'd like to close with a medical detective story about kids. which was headlined here in Science as Sequencing Mysterious Cases Yields Mutations Linked to Seizures and Arrhythmias. And this goes back to Michelle's very kind and generous introduction about things that interest us and I think should interest people in an age. So what is SUDC? Well, it's SIDS in older kids. The only distinction between these two syndromes is whether the child is less than 1 or over 1. And most of us would believe that there is no watershed and mechanism that stops at 1 year and then begins at 12 months. In fact, it's a continuum, and genetics is confirming this idea. Allowing for its prevalence, maybe not as large as SIDS, but still a substantial subtraction of SIDS patients. In fact, the ratio could be something like 3 or 4 to 1. The number of cases is less for SUDC, but the number of funding, the ratio is practically infinite. And the ratio of publications is also extremely high. So if you normalize things according to the number of patients, SUDC is underrepresented. So how do we approach this? Well, someone who is both a researcher and a mom who suffered a tragedy in her own family started a consortium called the SUDC Registry and Research Collaborative. And she teamed up with a neurologist who was our colleague working on CBD as well. And they reviewed the kid's death, did pathology and genetic analysis, made a case report, and report these in clinical journals to use this information to identify at-risk individuals. And they teamed up with people in pediatrics at Columbia who had a genetic background and people out in the community at the Mayo Clinic who were involved in doing the detective story about figuring out what genetic and environmental factors triggered SUDC. And they collected up to now 130 family trios where there is a deceased child, boy or girl, pretty much 50-50, and 2 parents who were willing, in the wake of the tragedy, to give blood samples and where their blood samples could be compared with that of the child. And so that's called traditional analysis of trios, mom, dad, and son or daughter. And the exomes, the coding exons of the individuals are compared. And so this is the collection, and almost all the kids died in their sleep, and very few who were awake. And I'll come back to what happened while they were sleeping later. In most of the cases, maybe 40%, the kids had some sort of seizure history, febrile seizure, some other type of seizure. So that immediately makes you think about brain circuits. And this work was published a couple of years ago. So the exome sequencing of these trios led to tables like this. And this is just a fragment of a long list of tables where they were able to come up with candidate mutations that might contribute to the disorder. And sometimes they were fortuitously found mutations that were already described in the database as potentially being pathogenic. This is a description varied on, produced by a database. And they have numbers, they have various ways of cataloging them. And so we use this as a way of educating ourselves into how geneticists approach things, taking this as a test case, a tragic test case, but one that offered opportunities. And we compared the effects of synonymous variants and non-synonymous variants, variants that did not change the amino acid that was coded for, or non-synonymous variants that could, for example, change a glycine to a serine. And Matt Halverson, a very talented geneticist, with great patience to explain genetics to electrophysiologists, did the analysis. The synonymous variants provide a great control because there's been a change in the DNA, but no changes in the protein. We're not expecting these to be lethal. And in fact, we found that in a subset of genes which are labeled cardiac epilepsy genes, not distinguishing between disorders of electrical activity, whether they be in the heart or in the brain, we lumped them all together. We had 137 genes, and we looked to see what were the odds that you would find a de novo mutation in this subset of genes. And in fact, we found that the non-synonymous variants were, in these particular genes, were significantly affected at the 0.00032 level. So these genes are a natural place to look because they show the signs of what you'd expect if they produce disease. The control of having the synonymous variants is very satisfying because these were not significantly changed. And as you'd expect, because they're not changing the protein. Now, let's take these genes, take the mutations we found in the subset of 125 individuals, and then run these against other diseases of kids. Cancers, not expecting any effect. Other rare diseases, asthma, developmental delay. In all these cases, there was no enrichment of the genes in these diseases in our subset. But if we looked at autism, where there are 717 genes implicated by the Simons Foundation and other organizations, we expected to see an overlap of about 4.5 in 4.5 cases, but we saw in 11 cases there was an overlap, an enrichment that a highly significant p-value, telling us on genetics alone that the diseases that come up in SUDC are highly relevant to autism and vice versa. And so this is the type of analysis you could do when you're not actually looking for something. You don't have a predetermined hypothesis. You're just taking what is given to you by people coming into the registry. So that's the conclusion, and it confirms what many people have always believed, that autism and epilepsy have a strong overlap. Not autism and heart disease or cardiac arrhythmias, autism and epilepsy. Shahan Wang took a different approach. He looked at the genes that were characterized and put them into protein-protein interaction networks. So people catalog the ways that proteins interact with partners by doing real honest-to-goodness biochemistry. And they find that the genes that we found affected, like CACN A1C, RYR2 in 2 cases each, and calmodulin, are all actually part of a social network, a network between encoded proteins that physically interact with each other. And for synonymous variants, this enrichment value was practically a chance, whereas in the non-synonymous protein-modifying variants, the likelihood of there being this much protein-protein interaction was highly significant. So not only do the proteins cause lethality, but they cause it in protein-protein interaction networks. And this was not only true of a cluster known for its role in calcium regulation, it was also true in a cluster known for neural development, which is very interesting because we were not expecting this. We were looking for something that would act acutely. This says that some of the genes we found clustered together because of protein interactions that were important in developing the nervous system. So what are the effects of these genes? Well, some of them are already known. The mutation in calmodulin causes slower calcium channel inactivation and is lethal with beta-adrenergic stimulation in mice. The renin receptor mutation caused a change in the calcium transient and a slowing of the calcium transient. I'm not sure what that means, but there's a definite phenotype on calcium release in cells. And finally, one of the other mutations that we found in our data set had been previously reported in a syndromic form of autism called Timothy syndrome, which is characterized by long heartbeats and a tendency to arrhythmias. We characterized ourselves, and this is Gary L. Grant with patch clamping, the time course of the L-type calcium current, and she found that there was a significant prolongation of the time course of the barium current in either of the two mutations which appeared in our data set. This is a common feature of mutations that cause arrhythmias and possibly changes in the nervous system as well. Highly significant increase in how much current there is late in the voltage pulse, as though voltage-gated inactivation is greatly slowed by the mutations. So putting it all together, we think that longer action potentials create a longer calcium flux. A longer calcium flux produces early after depolarizations and delayed after depolarizations that are a textbook example of how you can produce lethal arrhythmias. And it's both amusing and sad for me to realize that I've worked on this for a better part of 3 decades, going back to my work on heart tissue that Michel Antoine alluded to. Meanwhile, a group in Boston Shorelands published only a very short time after we did, independently, different data set of patients, and came up with a very similar conclusion that ion channels and other proteins involved are involved, and they you could take the two data sets together, and there would be both event overlap and an extension of the work. And because this is a relatively rare disease, it's an example where scientific cooperation could be very fruitful. Just because heart is an obvious possibility doesn't mean the brain is not involved, because the very same proteins are thought to be instrumental in excitation transcription coupling and other signaling abnormalities in different CNS disorders like seizures, migraine, and autism. And in fact, the L-type calcium channel conveys a conformational signal that doesn't need calcium influx, just by the protein conformation that could signal to the ryanodine receptor or to other proteins like KMK2, and thereby produce abnormal electrical activity. So the jury is still out about whether it's a cardiac disease or a neuronal disease or both. The heart disease could kill you because of an arrhythmia. The neuronal disease could cause epilepsy and an effect on breathing. And Laura and Oren have published a paper in Neurology taking videos of kids in their beds recorded as part of the normal surveillance that parents impose. And after careful viewing by clinicians, independent reviews, they concluded that there were a substantial number that showed terminal convulsions. Extremely sad for the families, but informative for how we can understand and cope with the disease and prevent, ultimately find a way of preventing it. And these are pictures of 3 of the kids whose mutations are actually known. There are enough of the mutations so there's no problem about confidentiality. And these pictures were provided by the families as part of their fervor to try to gain more public support for the idea of studying this devastating disorder. So to finish, and I'm going to do this in 4 minutes, we've been focusing on rare variants who have huge effects, as extreme as an autosomal dominant effect in causing a fatality. And these have huge effect sizes, but obviously they're very rare in the general population. How about the bigger public health problem of other variants which cause other diseases, neuropsychiatric diseases principally, which are extremely common, affecting a large number of people, but where the individual variation causes a small effect? And I'm going to sort of skip through this and say, we know a lot about the etiology of these rare mutations. How can we study the poorly understood ones? And one answer is we team up with geneticists who tell us in an unbiased way that there's a strong association with the CACN-A1C gene, the very gene that causes autism or causes SUDC. That gene is also important in other diseases that affect millions of people, including bipolar disorder, including schizophrenia, where CACN-A1C is the second in getting to the top of the charts. The same locus has been associated with major depression, ADHD, and ASD. So it seems that this important protein is actually not only important for normal function, it's important for disease as well. To be very brief, the genome is mostly non-coding RNA, non-coding DNA. The DNA is assembled by splicing, which puts together a mature transcript, which goes out into the cytoplasm and is used to encode protein. However, those instructions only use about 1% of the real estate in our genomes. 1% of our genomes are actually encoding, giving specific instructions to make the amino acid sequence in the protein. The rest of the genome is non-coding. What's it there for? It's to regulate the encoding parts, either by making the chromatin inaccessible or accessible, or allowing various transcription factors to bind. And to skip to the very end, we are focusing on TEC and A1C because about half the length of the entire gene is made up of one intron, and that intron harbors the SNPs for schizophrenia and the other major psychiatric diseases, even if you narrow them down to ones that are, by Bayesian analysis, thought to be causal. You still have 17 individual variations, but we're taking a frontal approach to this, led by an intrepid mountain climber named Shahan Wang, and we're trying to actually make artificial alleles which contain all risk or all protective variants of this, doing it by clever leveraging of molecular biological techniques. We hope to put these into human cells to study organoids and put them into mice to study behavior, and hopefully then we will be able to use the same type of approaches we've used for many years to analyze the basis of the disease by literally comparing what happens when you have an allele that makes you perfectly protected from schizophrenia or perfectly susceptible to it. We're not expecting all the risk factors to add because some of them are linked with each other, but we expect cumulatively will have a big enough effect that we should be able to see either a behavioral change in a mouse model or an expression effect in a derived human cell. So to conclude, genetics provides valuable impetus for neurobiology in connection to function. And SUDC is a really clear example of that. We had no idea where to look. Genetics told us where to look. But when we look, we find that the function of proteins is drastically affected. Dysfunction gives perspective on the healthy brain. Even in the healthy brain, there's LPI. It's being made all the time as a result of the action of PLA2 and LPI-AT1. And it's important in setting the EI ratio even under basal conditions. So when people talk about EI balance, which is 1 point along an EI ratio curve, ratio axis, that's presumably being affected by LPI. Who controls the controllers, like the excitatory, inhibitory synapses, calcium channels? Gene expression studies could focus on the signaling proteins that lie upstream of those signaling proteins, proteins like GPR55. The LPI GPR55 system follows the precedent of endocannabinoids and their receptor, EC1, studied here at NIH, among many other places. And despite all this intense effort, the lipid signaling still holds many mysteries because a lipid is not as easy to study as a neurotransmitter molecule, which is easily soluble and bottled and made into a dry and easily put into solution. The lipid doesn't like to be in solution. It likes to be in the membrane. So how does it get released? And who releases it? And how much of it is there? And how is it taken up? Inhibitory neurons are also controlled by fast inhibition. That's another one of Simon Chamberlain's loves. I didn't cover that today for the sake of time. There's a convergence between the genetics of LPIT and the pharmacology of phytocannabinoids. I gave you a genetic introduction, but actually CBD came about because a child, Charlotte Figge, had intractable seizures and her parents moved to Colorado to get some access to hemp plants that they thought would be viable. And that's what really lit the fire under the FDA to look into this and for people like Davinsky to run the unbiased trials, which are much better than anecdotal results. You need a large number of patients. You need to make it sound in order for the FDA approval. But they managed to get all the way from one patient and one brave family all the way to a drug. And finally, the importance of non-coding regions. This is the frontier of neuroscience as well as many other fields. And it behooves us to find ways of teaming up together and studying the regulation of the regulators by the non-coding regions of DA. I'd like to thank you guys very much for your attention. It's a little strange to be here at the heart of science in the U.S. at least, among people who are doing such great work. I have a lot of respect for the pioneering studies of people studying interneurons, studying cannabinoid receptors, studying signaling in many forms, really studying how the brain works. And it's a pleasure for me to be here and talk to you today. Thank you.
A little past hour, but I welcome a question from the audience. There's Dr. Owens.
Hi. Have you looked at the effect of high levels of phenylalanine on the GPR55 circuit?
No, we haven't. If you have a reference for me, please send me an email.
No, it's just a personal observation of someone who seemed to have phenylalanine-induced seizures.
Oh, really?
Yeah.
Okay. That's not something I would have expected, but thank you for that information.
In your model circuit, it only had positive feedback. So in the LPI circuit, not every hippocampus is prone to seizures. So what do you think happens in a healthy brain to tamp down? Are there endocannabinoids that act on this system?
There's a system parallel to the LPI system called the LPA system, lysophosphatidyl acid. So it has a phosphate group where the sugar ring, the inositol ring, is. So it's lysophosphatidyl without the inositol. Reminds me of Jack Nicholson. I'll take a chicken salad sandwich, hold the chicken, hold the lettuce, hold the mayo, because he wanted some toast. So LPA is the stripped-down version of it, and it has the effect of downregulating both excitatory and inhibitory transmission. So if you have a mix of LPA and LPI, you could have a system that could dial you up to anywhere you wanted to be in the coordinates. It's like my going to work by going east and going north, or going north and then going east. And so those two systems are the bare minimum that's needed in order for lipids to dial up whatever strength of E and I you want. Now, LPA is known to downregulate both excitatory and inhibitory transmission, but it hasn't been studied in the same tedious way that we did it to figure out what makes it release and how strong is it, and is it part of a feedback loop. But that's the negative component. So LPI is there to make the hippocampus live dangerously, live near the edge. You know, put blindfold on, put your foot down on the accelerator, take off the brakes. But there has to be a counteracting to that. And I think it could be LPA, or it could be an endogenous cannabinoid and acting in the way that you're alluding to. And so the combination of how these work together is still uncharted territory. Thanks for the question.
Okay. Thank you, everyone, for coming, and please join us for the reception in the hall, in the lobby. Goodbye. Thank you.